name dengan metric number so that I can respond accordingly lah ya. So siapa yang tanya pasal air tu ya? Tanya pasal air.
Saya doktor.
Shakira. Okay. Did you read my answer?
Dah, tapi yang artikel Prof bagi tu saya belum sempat go through lagi sebab baru perasan malam tadi.
Okay. Tapi ada lah ya. So maybe a lot of you nampak I posted an article as well. So sekejap ya. So saya akan share my screen ya. Macam biasa. Are you all ready? Yeah. So today we will look at toxicokinetics ya. So boleh nampak ke my screen? Boleh ya?
Boleh.
Okay. I'm just going to kecilkan ni, alright, so that I can see your messages. Yeah? If there are things that you want to ask me, alright. So ini yang I bagi tahu. Yeah? I posted an article, if I'm not mistaken, on the categorization, or another way, a different way that people actually class toxic substances. Okay. Sebab Shakira tanya, mana soalannya ni? Air, water. Yeah? I don't suppose water, if it's not contaminated, is a toxic substance, Shakira. But of course, the way that it's being ingested, if it's too much, and what or how it can cause the overall systemic effects, so that could perhaps result in a toxic punya manifestation lah, Okay, so I think you can read lah my answer. Hopefully you can understand as well. So boleh Shakira, mana you tak nampak dah?
Boleh Prof.
Alright. Okay, thank you. So I'm hoping this class tak panjang ya. Every time I akan cakap macam tu lah. But it's absolutely critical for us here in the campus. Tak elok lah pagi tadi I buat kelas berkali-kali kena cut off. So I suppose the campus is doing maintenance lah. So kalau saya cut off or suddenly I'm logged out, hopefully you all be patient. I can cuba masuk balik, boleh ya? How are your internet connection dekat rumah? Okay ke?
Okay.
Boleh ya? Alright. Okay, very good. So I try my best lah ya, not to be too long with you doing synchronous. Tapi tadi I rasa, or perhaps I hope I tak block one of you to come in. Sebab bila I click tu suddenly one of you not allowed to join in. So kalau ada, hopefully minta join semula and then I will admit. Eva, tolong ya. Tak ingat saya siapa tadi. Okay, so we siapa ya? So hopefully she will message you ke? Sebab ada dah. Okay, dah masuk dah. Okay, hopefully lah ya. Alright, so very good. So ada nampak ke rubrik dalam Padlet ni? Did you all see? Nampak ya? Ini simple rubrik saja. Yeah? Sebab the last time I was using a rubric yang I don't know where I got it from one of the other lecturers. So complicated sangat. So I do not think also the rubric was fair. Sebab ada weightage lah kali dengan markah lah. So hopefully the simple rubric will guide and assist you. So just now I think Eva also tanya kalau present tu meaning no need to present. Apa dia Eva? Apa dia tadi? No need to present apa? No need to present your opinion. opinion and concern.
Suggestion. Yes.
So kenalah present opinions and concerns, yeah, because if not, your presentation will be hanging, yeah. But of course, I wanted the write-up as well, yeah, so that I can see on how you approach the subject matter in the form of a written narrative, yeah. Hopefully you can make it work sebab ada 4 group saja kan, betul ke? 4 group, betul. Betul ya, 4 group ya. Am I correct, students?
Yes.
4 group. So dalam satu group ada ramai ya. Just so that you all get some work to do lah. So mungkin ada yang fokus on write-up, mungkin yang akan buat presentation. So I'm all for any form or presentation in any format. So don't worry so much. Yeah. Ada apa-apa lagi ke nak tanya about the assignment? Kalau you are not able to use your mic, you can chat also with me. Okay. So you are okay with the assignment? Okay.
Okay. InsyaAllah.
Ada orang ke tak ada orang ke tengah makan lunch? Okay, thank you Nora lain. Alright. Okay. So hari ni kita just do this. Yang inilah, I think yang ini ya. Okay. Alright. So just Toxicokinetics ya. Kalau tak salah saya, at the end of the last lecture, there are some slides that we actually did not go through. Tapi if I'm not mistaken, it's all in here also, right? So I think we can do just this lah. Alright. So ada apa-apa ke nak tanya sebelum saya mula? Ada important points ke that you'd like to ask? Sempat ke review? Tak ada. Tak sempat review? Senyap saja. Did you do your review?
Sempat tengok separuh je doktor.
You mean the last session?
Tak yang ni.
Oh yang ni, okay. Yang ni pun tak apa, but the last session, hopefully you have gone through the video. Dapat ke video tu? Somebody asked me whether or not they can display somewhere, play somewhere. Playlist. You all have playlists kan, yeah, for your batch, yeah? Am I correct?
Yes, Dr. Prof.
Playlist apa tu? Macam video, collection of video recordings ke? Yeah. Okay, tak apa. So, okay, thank you. So, hopefully you all tak disseminate ya. The video is just for you all lah. Segan saya nanti ya. Alright, so kalau tak ada issues ya, or perhaps later after this pun you can again review the last week's session. Then kalau ada just simply tanya ya. Okay, so you can put your questions in the dashboard in the Padlet ya. So I will attend to you there ya. Okay, so let's see. When we are looking at toxicokinetics, I think by right or by now you would understand lah. Macam pharmaco kan ada pharmacokinetics, pharmacodynamics, yeah. But since we are focusing on substances which can cause toxic effects, so we are looking at toxicokinetics now, and also toxicodynamics lah later on, yeah. So the principles are the same. If you're talking about kinetics, macam pharmacokinetics tu apa dia? The broad definition apa dia kalau pharmacokinetics? Ingat lagi tak?
White body does to the drug.
So the same, yeah. Okay. So maknanya you should be able to explain about absorption, and then apa tu distribution, biotransformation, and also excretion ke elimination ke, yeah. So this following the same principles, because anything that enters into our body, it will undergo all those processes, yeah. But what can you just briefly tell me in terms of benda yang kita belajar hari tu, route of administration kan, apa perbezaan dia dengan sekarang toxicokinetics, route of administration? Is it a bit different than previously? Kalau the
Yes.
toxic ni, okay, but how so? How different? So kalau now you nak kena describe the apa ni, how the toxic substance gets into the body. So how will you be describing this, the process or the event or the incident? Selalu kita administer ke? Selalu kita administer ke toxic substance pada orang?
Tak
Okay.
expose.
Yes, very good. Masuk temporary, okay, boleh juga. Tapi dalam exam jangan tulis macam tu ya, Niana. Alright, so yes, macam accidental exposure ke accidental administration ke, but it's usually without apa ni or perhaps without us knowing, yeah. So itu perbezaan dia. So you have to be careful when describing this, yeah. We don't purposely administer toxic substance for testing, in especially lah, yeah, in human beings, yeah. So it's usually having to do with exposure. So remember what I'm saying, yeah. But of course, toxic substances can come in along with apa ni, substance that we use for treatment, medication kan, yeah. And then also things that we eat, yeah, for nutrition purposes, yeah. So we can have toxins in there that we can digest, yeah. I mean we can ingest. So that can happen. And then also you have to think about dose, yeah. So ingat lagi tak apa Paracelsus apa ni cakap the last time, did we talk about Paracelsus?
Yes.
There is like a very thin line between dose and remedy kan, yeah. So you see, all those perhaps you are able to understand and able to articulate, yeah. So kalau you tak faham apa saya cakap ni, you just tanya, yeah. Okay, so that is very important lah, yeah. So itu kita nak didik lah you all, yeah. So that you understand something and then you would have your own views and you're able to like tell others, all right. So try to pick up that skills, yeah, along the way, right? Okay. So okay, so I think we can go through lah, yeah, on some of the important points. So I'm trying to actually take you back to dose response relationship lah, yeah. So this is also something very important when we are learning about toxicology. It has also been put forward by Paracelsus himself, saying that experimentation is very important, looking at dose and response, yeah, cause and effect, very important, right? So here we can come we can actually get many types of responses, yeah. But the usual one are these, yeah. graded dose response or quantile dose response. Yeah. So if you are looking at this, this is I think is very familiar to you. Okay. So meaning kita bagi dose banyak ni, we get this much response. If we increase the dose, we get maybe a higher response. Yeah. So the general rule is like this lah. Yeah. So the more that you give, perhaps the more response that we can see. So I think you also know this is described as a sigmoid type of graph. Yeah. And then learning toxicology and also learning pharmacology, I think you know about this. Yeah. There is that point whereby there is like no observable adverse effect. Yeah. That is the NOEL, the unit that sometimes toxicologists have to use to explain dose response relationship. Yeah? So meaning that is actually the highest dose that failed to cause the endpoint measured. Yeah. Uh, Selepas tu kita tinggikan lagi sikit, barulah ada observe adverse effect. Yeah, but sometimes it's not so easy to get this NOEL, right? So sometimes tak jumpa. Okay, boleh faham ke? Boleh ya? You can chat with me so that I can know that you are listening. Yeah. Okay, so hopefully you can follow lah. Alright, so quickly, here is the type of all or none type of response. Yeah. So yang ini, so this is the quantile punya type of response. I think you know about this. Normal distribution. Let's say kita buat kajian dalam satu-satu population. Yeah. So response terhadap inhalation of a toxic substance yang terlepas from a factory ke apa ke. So ada orang yang akan fall sick, ada yang tidak, ada yang resilient ya, tapi ada yang susceptible. Okay. So the correct word is here lah ya. So ada yang akan hyper susceptible ya. Sikit saja pun dia akan jatuh sakit ya. Or perhaps get some sort of illness kan. But there will be those here yang very resistant. Only when exposed to like a high dose will they like fall sick. But this can also be a dose response curve that actually shows mortality perhaps here or tumor production. So I think you can get a lot of literature writing about dose response like this. So there is a way of classifying according to the stats of the response, right? Okay, so itu just like the types of dose response in terms of the variation that you can see. Yeah. Okay, have we talked about probit the last time? Probit tu apa? Probit. Sebenarnya itulah dia probability unit. Yeah. Okay, so it's usually to do with mortality lah. Yeah. So sebenarnya let's say we get that sigmoid curve. Yeah. So we will turn it into a log dose curve, turn the curve into a line, so that we can actually describe what is happening much better. So here, so I think you can see here, like this is a log probit scale, so that it allows for you to actually, or perhaps toxicologists to actually describe the LD50 according to LD50, according to LC50. So data is very much linear. So as for LD and LC, I hope you remember from the last time. So you don't forget. So have we looked at this? Kita dah tengok ke ni hari tu?
Yes.
Kalau mengantuk tolonglah makan apa-apa. Saya pun belum makan lunch lagi ya. So you stay alert ya. So I'll try to be very fast lah ya. Okay, so yang ini is actually made linear for you to for toxicologists, toxico people, or perhaps you as a student of toxicology to be able to interpret and extrapolate lah ya. And then of course there is that confidence, the curves is actually representing the confidence interval lah ya, whether or not the results are reliable. Okay, so I think I've asked you which chemical is safer to use, and you said A. Yeah, so betul lah tu ya, because as we go higher in terms of the exposure of the to the toxin, yeah. So the response naik sikit lah daripada yang B. Okay, so B we can get things can turn fatal quite quickly. Yeah, so perhaps here you can straight away see A is safer than B. Yeah. So yang ini I think you know lah ya. Okay, so these are like how you can compare those responses. Yeah. Okay, so ED is all here lah ya, effective dose, toxic dose, lethal dose ya. So that is usually how it actually manifests. Perhaps medication that we use as treatment for treatment so effective dose at this much but as we go higher we can see that we can now get toxicity and it will turn out to be fatal in the end, right? So okay, so kita boleh kaitkan juga dengan how potent or how efficient the toxin can work so kalau macam ni, macam mana nak describe ni A dengan B ni, tell me anyone? Kalau A dengan B, so maknanya simply if it's potency, you can see here lah A will definitely be more potent. So that is how you would describe it. Tapi kalau C dengan D sama juga because C at a lower dose than D, it can also give you that toxic response for instance. Okay, but there is that efficacy issue. So here kalau A sama saja efficacy dia, but kalau C dengan D, you can see that C plateaus at a certain dose so D maintains. So you can simply say D is has a higher efficacy lah than C. Okay, so are you picking up points there? Ada apa
Prof,
nak tanya ke? Yeah.
I don't really understand why the potency for the C is meaning that C is more potent than D.
Sebab dia boleh act at a lower dose than D. It can give the same level of toxic response, for instance.
Okay.
Kalau macam ni, faham ya? Okay. So kita boleh macam sum up lah generalize that kalau dalam case macam ni C is much more potent. Dapat Okay, so get that point So do you see so those are like how you can describe dose response relationships. So you boleh guna potency, you boleh make comparisons in terms of the efficacy of two toxic substances ke, for instance so apa yang kita nak ajar ni just how for you to I mean perhaps to get your understanding and then to help you to be able to describe, to explain. I think ada ni dalam you punya learning outcome. So most likely your learning outcome will be to explain things, to be able, So if you are able, then perhaps I've achieved my teaching and learning punya outcome lah ya. So that'll be very good. Boleh faham ya? Okay. Alright. So for the others, yang ini I think I've actually mentioned to you. So now we go into toxicokinetics proper, but now we really look at toxicity punya definition. Yeah. So how is it that you can explain about toxicity? Yeah. So there are a lot of things that you need to consider. Yeah. But I think you know this, yeah, from what you learned in pharmacology before. Yeah. For instance, at the portal of entry, yeah. So concentration of the drug, of the toxic substance, yeah, and how long is the exposure. Semua tu akan be translated into how severe the toxicity is. Yeah. And of course it goes on lah, yeah, how much is being absorbed into the biosystem, yeah, and then sama ada cepat ke lambat ke absorption ni, yeah. So it usually goes by this way, yeah. So kalau a highly toxic drug that is poorly absorbed, yeah, kalau you bandingkan dengan low toxic drug for instance, tapi absorption dia cepat, so the level of toxicity or the manifestation of toxic effects that you can get could be the same. Yeah. Okay. So think about this. Yeah. So those are what will determine toxicity. Yeah. So it continues from there. Yeah? Sebab you ada apa tu selepas exposure tu kan ada A, D, M tu metabolism, or I like to use biotransform lah ya, and also elimination ya. Okay, so continues from there lah ya. Then you can talk about the distribution of the toxic substance, kat mana dia pergi. Of course lah, you can expect they to be like easily go to highly perfused organs, yeah, like the brain, the liver, the kidney. Okay, so sometimes only certain organs are affected, yeah. So it depends, yeah, on the type of toxic substance that enter into your body, and also the cells or target areas that it can affect. Okay, so I have included one example here, macam organochlorine compounds, ya. So sangat bahaya kalau apa tu dia lodge or dia deposited into adipose tissue, ya. Tapi sebenarnya target organ ni could be the brain because of the receptors ke. Okay, so that is how or perhaps what happens, yeah, when the drug or the toxic substance gets distributed in the body. You faham ke tak ni? Yeah? Tanya. Are you all listening? Can you all follow?
Yes.
Yes.
Alright. Good to hear. Okay, so and then you continue ke biotransformation. You know kan, sometimes parent molecule, parent drug, parent toxic substance, bila dia metabolize, suddenly the metabolite is much more toxic. Yeah. So it depends, yeah. And then of course how the drug is actually going about in the body, right? So of course whether or not the toxic molecules can be deposited into our body, it depends on the excretion, yeah. But I think sometimes I think you know that sometimes certain toxic substance can actually be stored, yeah, in your body. For instance, apa I tulis sini, lead, yeah, in bones, yeah, is one example. Okay, so for all you know, you could be accumulating certain things, yeah, in your body, yeah. Okay, so that semua needs all these aspects can contribute to the overall toxicity. Okay, yeah. So that is perhaps how you would explain toxicity in relation to toxicokinetics. All right, yeah. So hopefully boleh faham lah ya. Okay, so I think you can go through this or perhaps tengok ni nanti sama saja macam hari tu ya. Ini pun sama sajalah. But what is different here? Okay, so have a look at toxicokinetics dulu. Okay, so alright ya. So exposure ya. And then ini sama sajalah ya, sama saja concentration in the medium and then masuk dalam biosystem ya. Medium ni could be in your air, in the water that you drink, yeah, contaminants. Boleh dapat okay. And but then it enters into our system, yeah. So it will be in some level of concentration, and then as you know, the body will bring it to other system circulation, for instance. Yeah? So what is interesting when you go on to actually dynamics, yeah. So jangan ingat badan you ni biasa okay. Boleh dengar ke?
Boleh.
Okay.
Boleh. Sorry. Pernah je bercakap, tiba-tiba hilang. Yeah. Okay, so hopefully I'm clear now. So I do not know how often that will happen, yeah, but I'll try to go through very fast lah kalau macam ni ya. Okay, so coming back to toxicodynamics, yeah. If you can have a
I'm not presenting.
okay. Oh, kalau dah terkeluar dia ni lah ya, kejap ya. Okay. Okay. So nampak ke my screen? Nampak ya, okay. So, alright. So chat with me if you have any issues. Okay, so I'd like to focus on this damage recovery. Okay, so your body is great. Allah has made it that way lah. So there will be damage recovery. Yeah, and then of course the redistribution of energy. Yeah, and then how we respond lah. Yeah, so sometimes the damage is irreparable. Yeah, so it depends lah. Yeah, so perhaps there is the resulting pathology, yeah. But of course, whether or not it can be reversible or not, yeah. So that is following exposure to toxic substance lah, yeah. So as you can see here, this becomes a very important slide lah, yeah, for you to understand. Ada apa-apa ke nak tanya yang ini? I think dia macam logic lah juga lah ya. Okay, but for you to be able to actually describe, I would like for you to actually read around the area, yeah, to get some points, yeah. so that you have an answer for this. Yeah. So what happens during toxicodynamics? Yeah. So kan masa dulu kita belajar pharmacodynamics is basically what the drug does to the body kan. Yeah. So dia punya mechanism of action, mode of action. Yeah. So the resulting effect tu apa? Yeah. So at cell level, at organ level, at overall organism level. Yeah. Apa effects dia? Yeah. So in the end, will there be reversible or irreversible pathology? Yeah. So hopefully you can read around the area to be able to actually describe and explain. Yeah. Okay. Boleh ya, students?
Boleh.
Are you all okay? Okay. All right. So okay, so yang ini pun basically the same lah ya. So nanti you have a look through, ya. Okay, so sebenarnya, okay. So I think I keep reminding you lah ya. Kalau macam toxicity study ni, you don't purposely ya, macam study or administer or expose human beings. Yeah. So usually safety and toxicity testing is being done in animals, right? So of course kita nak dapatkan the dose response, the relationships, yeah. So that we get data. So I think you understand also different species of animals, different types of results. Okay. And then is non-clinical lah. Bila clinical, maknanya kita dah test on humans, ya. So for toxicity study, memang kita test on animals lah, ya. But you are aware kan, ya? Even when we are like testing drugs, ya, for treatment in human beings, can we get toxic effects? Boleh dapat toxic effects tak? Brothers, sisters?
Macam mana, Prof?
Let's say ubat tu dah macam the drug, during drug development, dia dah undergo animal testing. Okay, so now kita nak beralih pada teruslah ya, clinical testing to use on humans ya. Tapi akan ada tak toxicity effects during the clinical trial?
Ada.
Boleh.
Oh, good, dia ada. Ya, okay. Sebab kadang-kadang tak boleh nak detect yang dalam animal study tu. Ya. And then dia dah lepas stage ni, dia dah pergi ke clinical trial. But of course sometimes we do see macam some people will be allergic, hypersensitive, some sort of reaction, or perhaps ada yang mati, remember? Yeah. So then the drug will be recalled. Yeah. So you see, so maknanya when we are talking about toxicity study, memang kita buat in animals. Yeah. Usually it's non-clinical lah testing program to see what sort of toxic effects semua tu, yeah. But of course, even when it has gone through kalau ini medication lah ya, even when it has gone through to clinical study, still kita monitor, ya. So there will be like continuous feedback with clinical studies. Ya. So ini bercakap pasal drug development lah ya, because you know drug yang kita guna untuk treat conditions or disease pun can have toxic effects, ya. So makin lama makin enhance lah ya the toxicological data that we can generate. So dia jadi macam database, data bank, so that people yang nak study or nak guna drug ni for instance, ya, they know what to expect. Okay, dah faham ke? Can you understand?
Yes.
Yeah.
Yes, Prof.
Right. Okay, so so yang ini I can go through with you in the following slides lah ya. Okay, so when you are looking or studying toxicokinetics, yeah, so there are principles that we have to know, yeah. Of course, first and foremost, dose and concentration in the target organ. So berapa banyak, yeah, how is the level to cause what sort of toxicity, yeah, how severe the toxic effects, yeah, and then of course the kinetics of a drug may differ from its therapeutic dose to its toxic dose. Remember yang Paracelsus tu, ya. Okay, so ini bercakap just simply kita ambil contoh medications lah, ya. Of course the study of toxicokinetics ni sangat berguna in actually predicting lah, ya, the plasma concentration of a drug, ya. The parameters are actually very important, right? Dalam toxicokinetics memang quantification is very important, ya. So how to quantify? Macam mana nak measure? Macam mana ya nak measure kalau benda dah masuk dalam badan kita? How do you measure? Kita ambil apa?
Darah.
Darah kan, ya? Okay.
Blood test.
Yes, blood test usually lah. Tapi kalau dalam animals, kita boleh dalam animal study kita boleh sacrifice and then ambil liver lah, apalah, but we cannot do that in humans, yeah. Okay, so the usually is like taking samples from human beings like blood or plasma, serum, boleh juga ambil hair, yeah. Boleh juga ambil urine, okay, so that we can actually measure using equipment, yeah, to measure the toxic substance that we can identify, yeah. So itu proses what we call as quantification, yeah. Kita can then do assessment lah, ya, or evaluation on how it affects any test species ke, ya, or kalau dalam animal, kalau dalam humans macam mana, ya. Dalam lelaki, perempuan macam mana, ya. Sangat penting, ya. To be able to quantify, to get quantitative data becomes very important, right? Okay, that is one principle, yeah. And then yang ini, I think I've actually talked to you about exposure, but here is about extent of exposure, yeah. Berapa banyak yang terekspos, ya? Sebab kita nak kaitkan dengan the toxic effects yang kita tengok, ya. So for instance, if I ask you about kan orang non-Muslims lah, ya, let's say minum arak, ya. Muslim pun ada minum arak juga, ya. I am taking example yang non-Muslims minum arak, tapi kan ada recommendation daily dose, let's say for in between male and female. Pernah tak you tengok the daily recommended dose for alcohol intake? Have you all do you all know this?
No.
Ya, iyalah, I mean kita tak ambil tahu kan, ya, but I think you would you can guess, ya, the daily recommended intake for ladies selalunya lebih sedikit daripada daily recommended intake for men, ya. Simply because how the female will process lah, ya, dia punya alcohol dehydrogenase ke different levels, ya, than men, ya. That is why toxicokinetics ni sangat penting, ya. Dia bagi macam estimation, ya, so that the public will know the levels that could be dangerous to them. Boleh dapat ke tak ni? Okay.
Boleh.
Ya, okay. Kan penting kan, walaupun kita ni juga yang belajar toxicology ni, but we are also like the public kan. We would like to know apa level sedut carbon monoxide ni tapi kita masih boleh berjalan tak pingsan, things like that, or what is the recommended or perhaps the advisable number of hours to be in the sun before you. That's why kerja toxicologist ni sangat penting lah that is why macam dalam radio for instance dalam radiology kan, ada tak you know about staff having to take radiation leave?
Yes.
Pernah dengar tak? Pernah dengar ya? Radiation leave tau, betul. Dalam borang cuti tu pun ada boleh take radiation leave. Ini untuk staff yang exposed to radiation so after a certain level of exposure, they are required to take radiation leave, cuti. So meaning calculation of the levels tu itu memang kerja toxicologist, radiation people, so it gives an estimation on what would be safe levels. Okay, so apa extent exposure tu yang masih safe for people, right? So I hope you can get that. Boleh so itu memang principles lah when you are exposed to things that can give you toxicity, toxic effects, dangerous or lethal effects. So I think we can move on. Okay, yang ini kalau nak quantitate tadi of course sampling points so that we can get macam the dose response ke but of course kalau macam toxicity testing in animals selalunya we wouldn't want to cause physiological stress for instance to the animals. What if the measurement tu sebenarnya nak measure hormone kan, kalau stress the hormone levels will be haywire so you have to be careful also when you are doing this testing or sampling, right? Okay, so of course let's say kita nak buat kajian dose response ni. So macam lecturers kan, your supervisors semua nak tulis FRGS, nak buat animal study. So they have to like read around the area, read on what people, other authors or previous researchers have done so they can find out the doses that they will use in this toxicity testing that they want to do. So that is very important actually. So kena setkan the dose level, right? So itulah kena consider or look what would be the lowest dose level used. So there needs to be consideration on the dose levels, right? So you can have a look here also. Okay. So other factors to be considered, of course, yang ini you dah belajar, you have that pharmaco knowledge. Ingat lagi tak protein binding? Can anyone tell me protein binding tu apa? Dah tak ingat dah? Bila belajar? Last semester kan anyone wants to chat with me? What do you understand by protein binding? Kat mana protein binding ni happen? Anyone?
Enzyme.
Protein apa yang kita bercakap ni? Protein. Ada ke orang petang-petang ni?
Plasma protein Pak Doktor.
Betul. Contoh macam albumin kan. Kan let's say toxic substance or drug ke masuk dalam body, dia akan masuk dalam systemic circulation. So some drug molecules ni dia highly protein bound. It affects the distribution in the body, kan ya? So of course lah ada effect on the resulting toxicity, ya. So tengok balik apa maksud protein binding, ya. And of course you have learnt about this under pharmacodynamics, ya, about tissue uptake, ya. Properties at the receptor, and then metabolic punya profile semua ni, ya. Semua ni I think you kena consider lah, ya, when you are about to embark on a toxico punya study, ya. Gunakanlah knowledge yang you dah dapat masa belajar pharmacokinetics, pharmacodynamics, ya. A lot of factors to be considered, ya. And then of course, kalau nak buat toxicity study ni, kena fikir route of administration, ya. Sometimes orang nak study the effects of inhalation of yang biasa tu lah carbon monoxide ke, smoke ke, ya, secondary smoke ke. Kena fikir lah, ya. Takkan nak inject pula rat tu dengan carbon monoxide. Kita kena macam simulate the reality, ya, dalam kita punya experiments, ya. Have you all seen macam experiments yang whereby rats are made to inhale I do not know, some sort of gas ke, ya, to see the effects, ya. So that the scientists can get a dose and response relationship, yeah. So of course route of administration pun something yang kena fikirkan, yeah. You have to establish lah, yeah. But of course semua ni I think you can get advice from the OECD website. Ingat lagi tak apa OECD? OECD. Anyone? Ada ke I cakap benda ni last last semester? OECD. Pernah come across tak OECD?
Tak sure Doktor Prof.
Ada ke?
Tak sure Prof.
Ini macam testing methods, testing protocols, ya. Ada dalam website yang Organisation for Economic Corporate Development ni, Economic Corporate Development ke, ya. Whereby dia dah siap dah macam methods, ya. Kalau nak testing of chemicals ke apa, ya, toxic substance. Macam dia dah siap recommend kalau untuk testing chemicals macam ni guna rabbit for instance, ya. And then kena guna female ke or dua-dua female and male, ya. Ada recommended methods, ya, that you can get from OECD. I suppose nanti when you go through with me about toxico punya testing, ya, towards the end of the semester, I will actually yes, yeah, cooperation, okay. Thank you Syami, ya. Ada dalam dia punya website ni, when you go to health, if I'm not mistaken section 4 akan keluar, ya, banyak sangat methods that we scientists can adopt, ya, and adapt, right? Bila nanti kita buat research guna methodology OECD, macam kita tulis publication kan, senang nak di-accept, ya? Because the reviewer will know that you have used foolproof methods, yeah. It's very useful to us actually, right? There are things like that will be advised by the committee under OECD. Malaysia belum masuk lagi, ya, belum member of OECD countries lah, ya. Rasanya macam belum lagi, tapi we are actually using, ya, the OECD protocols, ya, and guidelines, right? Okay, coming back to here, ini pun you kena fikir, ya. This is what toxicologists do. Dia kena fikir, dia kena macam emulate the actual happenings, ya, in real life kalau nak buat testing ni, ya. Kena consider or look properly at the route of administration. Okay. So ini macam contoh-contoh lah ya. So here we have intravenous, yeah, oral, okay. Okay, so the different relationships, dose response relationships that can result. And of course, you have to think about the metabolite determination. So maknanya, right from the very beginning also, bila you nak embark on this toxicity study, you kan tadi nak measure, nak quantitate, ya, you dah kena ready, what sort of metabolite that you will want to measure, yeah. So that is why it's very important that you read around the area, you decide or do the research or study planning beforehand. So the metabolite determination becomes very important in the study of toxicity, yeah, or toxicity testing pun, yeah. Okay, so I think in terms of metabolites resulting from biotransformation, remember we learned last time, ingat ya, macam when we have parent compound and metabolites, ya, sometimes metabolites ni sama saja dia punya activity, ya, dengan dia punya parent, ya. But then some metabolites dia lagi potent, lagi toxic in this case lah, since we're talking about toxicity kan, ya. Lagi toxic from dia punya parent molecule. But of course, what we are aiming usually during metabolism tu, body memang aim untuk actually produce a metabolite that is like less toxic already, yeah. Okay, so remember those points, okay. So metabolite determination pun sangat penting, and of course we make use of stats lah, ya. So those are like the principles lah, ya. Okay, so okay, so what methods to use? Selain daripada go to OECD ni, nanti I akan bagi link, ya, so that you can see the range of testing, toxicity testing that they have, ya. And then what sort of equipment or analytical methods that can be used, ya. So kan tadi macam kita nak identify perhaps the components, yeah, in the toxic substance, yeah. So perhaps we can make use of chromatography, yeah. So I think you are all very familiar lah, yeah, with all this equipment macam GC. Tahukah apa ni GC, HPLC? Have you all come across this? I know you are in year 2, tapi dah come across kan? GC is gas chromatography kan? You all have heard of this, eh? Okay, high performance liquid chromatography, semua tu lah, ya. So these are like useful equipment, yeah, machines, yeah, that can give you macam the levels of active components or toxic components in the toxic substance itself, yeah. Okay, so all these are like what to do, what to know lah, yeah. Okay, so ada apa-apa soalan ke before I go further? Boleh ya? Itu macam the principles lah, ya. Any questions you want to ask me at this point? Kita ada like 18 more slides saja. Yes, Nurul. InsyaAllah boleh follow?
InsyaAllah okay.
Boleh,
Boleh. Kalau okay, thank you. So kalau terlepas tadi, you look back at this video, yeah. Okay, so that you can understand lah, ya. Okay, so alright, so kita dah sejam. I think we are doing good lah. But of course, since we are learning about toxicokinetics, yeah, so memang tak lari lah daripada absorption, distribution semua tu, yeah. So I think you can go through this lah, ya. So prinsip dia sama saja macam what we learned in pharmacokinetics, ya. Of course, you have to be able to explain the absorption aspect, yeah. Here, of course, kita more concerned about exposures, yeah, especially when we are talking about exposure to toxic substance, yeah, for us human beings, yeah. So the rate and also extent of absorption pun sangat penting, yeah. That is one thing. Ni macam mana ni? Tell me. Please rate the following processes in order of fastest to slowest absorption. What do you all think? Oral, dermal, inhalation, intravenous. Yang mana yang fastest absorption into the systemic circulation? Tell me. Or you can chat with me. Boleh tulis. Nombor 1,
IV, intravenous.
confirm lah IV. Yeah. Terus je masuk dalam your blood circulation kan ya. So second will be
Inhalation.
yes, correct ya. Sebab in your apa tu lungs, yeah, there are a lot of capillaries, yeah. Straight away it can enter into the systemic circulation. So antara oral dengan dermal ni yang mana yang lagi slow? Which one?
Rahman.
Yang paling slow?
Dermal lagi
I
slow.
don't think so, yeah. Oral ni lagi slow, you know, because
Dermal lagi
dia kena lalu banyak-banyak hurdle, yeah, dalam GI tract kan, ya. Ada acid lah, ada apalah, ya. So dermal is much faster lah, ya, than oral. Okay, so think about this, yeah. So here we are talking about the rate of absorption lah, yeah. This is something also that you need to consider, yeah, when you are explaining about toxicokinetics. Okay, so this is another thing that I mean, just to give you a representation lah, yeah. So in instances where the absorption rate is lower than elimination rate, the rate of washout of toxicant becomes rate limited, yeah. So maknanya to hang on, to actually explain this to you, sebenarnya kalau macam the washout is lower, so maknanya the toxic substance dia duduk dalam badan lama, ya. So dia jadi macam depo, depo tahu kan, ya. So maknanya dia jadi macam reservoir. So dia masih distribute toxic substance to the whole body, ya. That is why washout is very important when you are talking about toxic substance in the body. Boleh faham ya? Okay. Alright. So meaning you should be able to explain in terms of the rate of absorption also. And also washout lah. Okay. This one you can have a look through. So ini contoh-contoh lah ya. Kalau absorption faster than elimination, so this is what we can get, ya. Kalau nak banding IV dose dengan non-IV dose, nampak ya, okay. And then kalau yang ini, kalau elimination faster than absorption, alright. Yang mana yang you nak actually? Kalau ada toxic molecules masuk dalam badan, which we would prefer? Students?
Elimination faster.
Elimination lah kan, ya. Okay. So that is what we aim for lah, ya. So let's say tercucuk kita punya muscle ni dengan drug, ya, yang not intended for us tapi kita nak administer dekat tikus tu. Tikus ni berkeliaran, tak boleh handle, ya. So we accidentally prick ourselves, ya. So that is like a non-IV dose, yeah. But if the elimination in our body is faster than absorption, this is what we are hoping for. Okay, boleh ya? Okay. So meaning kena fikir apa aim kita ya, kalau it's toxic substances that enter our body ya. Okay, very good. So remember we learned about bioavailability. Ingat lagi tak the definition for bioavailability? Anyone? You can even search and put here, yeah. It's essentially how much will enter the systemic circulation lah unchanged, yeah. So in this sense, systemic availability is also very important, yeah, when you are describing toxic exposure, yeah. And then dekat dalam badan kita ni ada tak barrier yang Allah dah provide. Let's say blood-brain barrier, belajar kan blood-brain barrier, yeah. So you have come across kan, yeah, dah belajar, yeah. And then of course the first pass metabolism by our liver, semua tu akan tolong actually metabolize the toxic substance, yeah. So how much is available in the systemic circulation will be very much depending on those, yeah. Okay, very good. Thank you, Shami. So okay, so this is something also that will determine the level of toxic substances in our body, right? So yang ini nanti you go through, ini cuma calculation saja. This is what we learned from previously, so sama saja. I think you remember this from previously also. And of course, bila you bercerita pula pasal aspek distribution, you can relate to blood flow. You can relate to permeability, yeah, kat mana the toxic molecules can enter or not, yeah. And then of course, the protein binding. Toxic molecules ni can also, ialah ya, dia molecules kan, drug molecules can also be toxic kan, so they can also be protein bound, right? Okay, so read about this, yeah. And the environment itself, pH, whether or not it has effects on the toxic molecules, and then of course the age of the person who has been exposed, yeah. So how it the constituents in the body will affect the concentration levels of the toxic substance. So kat sini, I take you back to the first and foremost point. Kan kita bercakap toxicity ni, it has to do with the dose kan, and the concentration at the target sites. Barulah mungkin akan dapat the manifestation of the toxicity, yeah. So let's say in old people, semua benda dah go down, the processes, okay. So even the metabolism dah macam dah tak bagus, So that is why the toxic molecules can be in high concentrations in the body, So that is especially dangerous, Then of course, we consider also where these toxic substances can go, okay. So they can be deposited in many compartments of your body, So can be in all these organs, adipose tissue pun, right? So hopefully you are listening, okay. Okay, so yang ini kita dah belajar kan ni? Have we looked at this? Did I teach you this? Ke the other lectures? Volume of distribution. I ajar yang ini kan?
Prof ada.
Students, can I get feedback?
Betul-betul.
You
Yes,
went through this with me kan? Ada ya? Okay. Okay, so I need not tell you lah about this, Okay, so senang kerja. Okay, so ini pun very peculiar for exposure to toxic substance, Okay, so macam ni, So let's say there is that bolus dose, one-time dose, intravenously. Yeah. So we can expect to get the graph curve to be like this. Yeah. This can also help you in terms of getting the volume for distribution lah, ya. But of course, kita balik pada prinsip yang dulu. Remember I cakap volume of distribution ni kan hypothetical volume, ya. So it very much has to do with yang formula ni, ya. It tells you whether or not the drug is like very fast distributed or very slow. Kalau drug tu masih banyak duduk dalam plasma sebab dia melekat dengan protein, so you can expect the VD to be, kalau C is high, maknanya VD you will get is low lah kan. So it tells you of how the drug is actually been distributed. Okay, so hopefully you boleh sit down balik and recover, remember what we learned from before. So these are all other representations lah, of how you can come up with the volume of distribution. Okay, so ini reason dia why it can be a hypothetical volume. Dalam badan kita manalah ada 50,000 liter ni. Okay, so yang ini I think ada tunjuk kat you dulu. Okay, so of course when we are explaining or we are trying to make sense about distribution of drugs or toxic substance, of course it will be determined by tissue or organ volume, and then some other, the drug can actually go through or pass through the organs. Here what is especially important lah, our aim is we hope lah, tadi kan we hope for washout, for elimination rate to be faster, and then of course we hope for macam reversible effects, so that kita protected from this toxic effects, right? So itu saja kali ni. So okay, are you all alright? Can you all follow? I'm trying to be very fast lah, takut I kena log out lagi macam tadi. Okay, so I think basically itu saja for now, But ada apa-apa ke nak tanya at this point? Anything you want to actually ask me? Ada tak clear tadi agaknya? Or you terlepas? Boleh follow, Nurul Ain boleh follow ya? Okay. Alright. Okay, so actually what it takes is for you to sit down. Kalau macam sekarang ni boleh study beramai-ramai ke? Do you do that during online ni? Where are you all actually? Kat rumah kan? Ada yang dekat rumah. Kat rumah So tak boleh buat group study lah. Boleh ke?
Tak buat pun.
Tak buat You don't organize macam Google Meet sessions between you all ke? Or perhaps bila ada assignment, then you need to do kan, get together, so try to also do revisions with one another, Okay, so perhaps then you can understand things better, But if every one of you macam tak boleh make sense ke apa ke, you can always ask me, So kalau tak nak apa tu, for Panadol, is it classified as absorption faster than elimination? It depends, I tak berapa ingat lah kalau macam Panadol ni. I think it depends on the individual. But remember Panadol ni dia akan follow first order kinetics. Ingat lagi tak first order kinetics as opposed to zero kinetics? Nurul Ain? Ingat lagi tak? Kita belajar kan ni dalam yang elimination. No, yang elimination lah, elimination. First order, you're talking about elimination kan? Kan ada a lot of drugs, a lot of things akan follow the first order elimination. Tapi akan ada macam a few things ya, macam alcohol, macam apa tu, phenanthroid ya, and apa lagi satu tu yang akan follow zero order. Ya. So yang lain-lain tu memang akan pass through our body ikut the normal elimination rate semua tu ya. But of course it depends very much on the individual. So let's say the individual tu ada kidney problem. So what can happen? Ada masalah tak nanti dengan elimination? Dia ada kidney impairment. What do you think, students? Or
Ada
anyone?
masalah.
Kalau ada kidney impairment, ada masalah kan ya, dia akan affect elimination ya. So things like that. Okay. So in terms of how it will be absorbed, sebab selalu Panadol kita makan, So kita tak inject kan Panadol. So it's usually oral ya. So it depends on you have to look back at oral route administration. So how much of the Panadol will be bioavailable. Sebab bila masuk dalam perut you semua tu dengan acidnya, dengan nak kena pass through membrane semua tu ya. So perhaps only a certain percentage can get into your systemic circulation before it can be distributed to target sites. Boleh ya? Boleh ya Nurul Ain? Are you getting me? Boleh faham ya? Allah, terus keluar. Terus connection ni ya. Okay, so the others you can understand
Yes, doctor.
Anything? Okay. Anything you want to ask? Ada apa-apa lagi ke? Yang you want to ask? Apa-apalah toxicity ke? Or you want to share your bukan toxic relation lah ya, tapi toxicity effect experience ke? But if not, think about this ya. So it's very much applicable to your daily life ya. Macam last week pun I ada cakap can we be absolutely sure that we are not constantly exposed to toxic substances kan ya. Even when you keluar, when you are in non-ventilated area ke let's say ya. Tapi kenapa kita tak jatuh sakit? So then you remember back ya. So what the body will do to the toxic substance. So the absorption, yang kita nak achieve tu the metabolism ya, and then the elimination, the wash out ya. So it's very much applicable to your daily life actually ya. So knowledge ni memang sangat penting lah ya. You use it sampai ke liang lahat lah. Okay. So ada apa-apa lagi kan nak tanya saya? Ada brothers ke dalam kelas ni? I've not heard anyone speak. How
Ada.
many brothers? Imad, I tahu tapi yang lain-lain tu macam tak berapa I've not interacted very much with the other brothers so hopefully later lah ya. Ada berapa brother dalam kelas ni Imad?
3.
Seorang tu?
Tak 3.
3.
Cuma seorang dia ada internet connection problem. Tu seorang lagi saya tak pasti lah sebab saya contact dia tak jawab.
Okay, tak apalah. So tak apa ya, because I will pass you yang recording ni ya. Okay. So as long as you have the will to learn lah ya. Sebab you know you have to do this kan ya. But it's good for you also this knowledge ya. So okay, so kalau tak ada apa-apa, can I conclude sekarang students? Boleh ya? Pagi tadi ada kelas tak? Pagi tadi ada kelas kan?
Ada.
Ada. Oh, letih lah, letih. Okay. Okay, so macam biasa nanti I will pass the recording to Eva or to the whole group lah. I hope you are able to view lah. Yeah. Okay, so manage your time well and make sure you understand the points that we actually went through just now. Yeah. So that senang ni nanti ya. So kalau apa-apa, you know where to reach me. You can also put your questions in Padlet. Okay. Okay, so I think we end our session lah ya with Ms. Farah and Surah Anas. Okay, terima kasih semua. Assalamualaikum.
Thank you.
Thank you,
Good
Prof.
afternoon.
Thank
ya.
you,
Good
Prof.
evening. Okay, thank you. Okay, bye-bye.
Terima kasih, Prof.